Assalamu alaikum. Our today's topic is about influenza virus. It is the most common and important virus for our viva, and sometimes we get it in our written question. So we have to know the important questions about it. The family of influenza virus, only member of orthomyxovirus. Then the types. It is the most important question. The types of influenza virus is three types: type A, type B, and type C. Type A, moderate to severe illness, antigenically highly variable, worldwide epidemic. Worldwide epidemic ke amra pandemic bole thaki. All ages group, humans and other animals can be affected by it. Then type B virus, milder disease, major outbreak, sometimes epidemic, primarily affect the children. Type C, mild respiratory tract infection, antigenically stable, no epidemic or outbreak, no gene for the neuraminidase. ইনফেকশন, ইনফ্লুয়েঞ্জা আমাদের ফ্লু লাইক সিমটমস ডিজিজ করে থাকে। এছাড়া এভিয়ান ইনফ্লুয়েঞ্জা বার্ড ফ্লু করবে, স্প্যানিশ ফ্লু 1918 থেকে 20 সালে প্যান্ডেমিক করে থাকে। সোয়াইন ফ্লু 2009 প্যান্ডেমিক, চায়না ফ্লু H7N9 2013 and recent H5N6 August 2021. Then the general properties. It is a single standard RNA virus, segmented genome, helical nucleocapsid, envelope outer lipoprotein. এখানে আমাদের একটি কোশ্চেন করা হয় যে লিপোপ্রোটিন থাকার সুবিধা কী। লিপোপ্রোটিন থাকার সুবিধা হলো এখানে লিপিড স্ট্রাকচারটার জন্য যখন আমরা সাবান বা কোনো ডিটারজেন্ট দিয়ে ওয়াশ করি তখন এই লিপিড স্ট্রাকচারটা দ্রবীভূত হয়ে ওয়াশ আউট হয়ে যায়, যার কারণে এই ভাইরাসটি ধ্বংস হয়ে যায়। তাই এই ভাইরাসটিকে ইজিলি ডিস্ট্রয় করা যায়। The another properties are spike of hemagglutinin and neuraminidase. Then we have to know what is genetic reassortment. When a cell is co-infected by the two different virus of a given type, mixture of a parental gene segment may be assembled into a new progeny virus, may result in sudden change of viral surface antigen. This phenomenon is known as genetic reassortment. Then we have to know about hemagglutinin. Hemagglutinin, binding of a virus to cell surface receptor, neuraminic acid and salicylic acid, neutralizing antibody against E, diagnostic value, hemagglutination inhibition test. Neuraminidase, cleave neuraminic acid, salicylic acid to release progeny virus, degrades the protective layer of the mucus in respiratory tract, enhance infection. অর্থাৎ এটি রেসপিরেটরি ট্র্যাকের যে মিউকাস লেয়ারটাকে আছে সেটাকে ডিগ্রেড করে, যার কারণে ইনফেকশনটা আরও দ্রুত এবং সহজেই হয়ে থাকে। দ্য ইনফ্লুয়েঞ্জা ইজ লুক লাইক দিস। We will face it in our OSP examination. We can easily find it by the hemagglutinin and neuraminidase structure. If it is present, then we can say that it is an influenza virus. Epidemiology. Pandemic occur every 10 to 20 years. Major outbreak occur virtually every year in various countries. In 1918, Spanish flu H1N1 avian origin. Pandemic kills World War I and II, Korean War, Vietnam War combinedly at least 500 million, one-third of the world population was affected, at least 50 million death. Antigenicity, group specific antigen, genomic ABC, type specific antigen, hemagglutinin and neuraminidase. Hemagglutinin and neuraminidase antigen changes with the time. This change causes devastating worldwide epidemic. There is two type of changes: antigenic shift and antigenic drift. It is very important for the viva question. Antigenic shift, major changes, new subtype caused by the exchange of gene segment, genetic reassortment. Entire segment of the RNA is changes. In our viva, we have to told this word genetic reassortment about antigenic shift. Examples of antigenic shift: H2N1 virus causes pandemic. H3N2 virus appeared in 1968 and completely replaced H2N2. H5N1 outbreak 1997 and H1N1 outbreak 2009. Another one is antigenic drift. Antigenic drift, a minor change, same subtype caused by the point mutation in gene, may result epidemic. Example of antigenic drift: in 2002-2003, a Panama 2007-99 H3N2 virus was dominant. A Fujian 411 2002 H3N2 appeared in the late 2003 and caused widespread illness in 2003 and 4. Here the Fujian is the type geographical origin, 411 is the strain number, and 2002 year of isolation, and H3N2 are virus subtype. The very important topic is clinical feature. Incubation period 2 days, range 1 to 4 days. Severity of illness depend on the prior experience with the related variant. Abrupt onset of fever, myalgia, sore throat, non-productive cough, and headache. Complication: pneumonia, Rey syndrome, and myocarditis. In pneumonia, secondary bacterial infection, primary influenza viral, Rey syndrome, encephalopathy, and liver degeneration in the children with influenza who are treated with the aspirin. Rey syndrome also very important topic for viva. Diagnosis: clinical and epidemiological characteristic, isolation of the influenza virus from the clinical specimen, nasopharynx, throat, and sputum. Significant rise in influenza IgG by the serological assay and direct antigen testing for type A virus. So this is all about influenza virus. Hope you like this video and it will help you for your examination. Thank you so much.